All right, this is Carbohydrate Metabolism. So we've got digestion. The types of carbs we eat are starch, sucrose, lactose, and cellulose. Starch is a polysaccharide carb consisting of many glucose monosaccharide units joined together by glycolytic bonds. This complex carbohydrate is similar to glycogen, as its main function is to provide energy. All plant seeds and tubers contain starch, predominantly as amylose and amylopectin. Amylose is a long unbranched chains of glucose linked at C1 and C4, represented as alpha-1,4 bonds. Amylopectin is highly branched polymer glucose, with glucose units linked by alpha-1,4 in a linear fashion, and alpha-1,6 at the branches, which occur every 24 to 30 glucose units. Glycogen has a similar alpha-1,4 and alpha-1,6 glycolytic bonds. There are three major classes of carbs: monosaccharides, disaccharides, and polysaccharides. Monosaccharides like glucose and fructose, disaccharides like sucrose, lactose, and maltose, polysaccharides, starch. Sucrose is a disaccharide made up of glucose and fructose found in table sugar and fruit. Lactose is a disaccharide found in milk, made up of galactose linked to glucose. Humans can only process sugars with the D conformation, or dextro right-handed. Think D for delicious disaccharides. Cellulose is a polysaccharide consisting of a linear chain, several hundred to over 10,000 beta-1,4 linked D-glucose units. Its primary structural component, the primary cell wall of green plants. Humans cannot digest cellulose because they lack cellulase to break it down. Salivary amylase breaks starch into maltose, which is made up of two glucose molecules. It's present in the mouth, where it begins digestion. It's deactivated by stomach acid. Pancreatic amylase, aka alpha amylase, facilitates starch breakdown in the small intestine, cleaving 1,4 units to release trisaccharides and disaccharides. Trisaccharidase breaks down trisaccharides into di and mono. Disaccharidase breaks down disaccharides into mono, glucose, fructose, and galactose. Sucrase breaks down sucrose into fructose and glucose. Lactase is secreted by the intestinal villi. It breaks down lactose into two subunits, galactose and glucose, for absorption. Some people with lactose intolerance might not digest lactose because of a lack of lactase. Undigested lactose remains in the gut and ferments, causing bloating and diarrhea. Glucose is absorbed and stored in the body as glycogen in skeletal muscle and liver. When glucose is needed, glycogen stored in the liver and muscle is broken down. The breakdown process of glycogen is called glycogenolysis. The breakdown process of glucose is called glycolysis. The glycemic index is a measure of how quickly blood sugar levels rise after eating a particular type of food. The glycemic effect of food depends on the type of starch, fat and protein content of the food, and the pH. Low glycemic index foods, GI less than 0.55, include beans, whole grains, most veg, sweet fruits. Medium glycemic index foods, it's a GI of 56 to 69, include pita, raisins, banana, whole wheat bread. High glycemic index foods, it's greater than 70, include potatoes, rice, cornflakes, bagels, and watermelon. Glucose transport. Glucose is found in the blood and needs to be transported actively from the blood to the cytoplasm of the cells. Glucose is the main fuel of the brain. Glucose is transported across various cell membranes using a variety of tissue-specific transporters named GLUT 1 through 14. The most important ones are GLUT1, RBCs, brain, placenta, testes. GLUT2, liver, kidney, beta cells in the pancreas. GLUT2 transporters are coupled with sodium, which assist in the transport into the cell. GLUT3, neurons. GLUT4, adipose cells, skeletal, cardiac muscle. Receptors are sensitive to insulin. All right. Glycolysis. The process by which one 6-carbon glucose molecule is converted to 2 3-carbon molecules of pyruvate. 2 molecules of ATP, 2 NADH are the net yield for each glucose molecule that's converted to 2 molecules of pyruvate. This is the simplest form of metabolism in the cells. It takes place in the absence of oxygen, so it's anaerobic. This is a cytoplasmic pathway used by all cells to generate energy from glucose. This pathway is also known as the Embden-Meyerhof pathway. I haven't heard that. Maybe I have. The purpose of glycolysis is it's the principal breakdown route of glucose to produce energy. It happens in all cells in the body. RBCs do not have mitochondria to finish the cellular respiration. The cell site takes place in the cytoplasm. Sequence of events, there are 10 steps leading to the production of the 2 pyruvate from 1 glucose. The key enzymes of glycolysis: hexokinase glucokinase, phosphofructokinase, this is the rate limiting step, and pyruvate kinase. There are 2 phases of the glycolytic pathway. Preparatory phase is the first 5 steps. The payoff phase is the last 5 steps. Preparatory phase is also known as the investing phase. Step 1. Glucose to G6-phosphate. It's catalyzed by glucokinase in the liver and hexokinase in other cells. This step traps a glucose within the cell as G6-phosphate, and it can't diffuse out. 1 molecule of ATP is used, and it's an example of substrate phosphorylation. It's irreversible. Step 2, G6-phosphate to fructose 6-phosphate. This step is facilitated by phosphoglucose isomerase. An isomerase is an enzyme that changes the spatial arrangement of a molecule without affecting its chemical composition. Step 3, fructose 6-phosphate to fructose 1,6-bisphosphate. This step uses another ATP. It's catalyzed by phosphofructokinase, or PFK. This enzyme is irreversible. This is the most important rate-limiting step in glycolysis. Fructose 6-phosphate to fructose 1,6-bisphosphate. Most important rate-limiting step in glycolysis. It is allosterically regulated enzyme. It's the major point of regulation for the entire pathway. Step 4, fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate. DHAP, we'll just say dihydroxyacetone phosphate, DHAP. This step is catalyzed by aldolase. An aldolase is a sugar which contains an aldehyde. So that's step 4, where we go to glyceraldehyde 3-phosphate and DHAP. Step 5, since DHAP is not desired as a substrate for continuing glycolysis, it's known as a ketone. Ketonone. It's converted to glyceraldehyde 3-phosphate. This step is facilitated by triose, a sugar containing 3 carbons, and phosphate isomerase. Triose phosphate isomerase is the full name. This reaction is an isomerization reaction converting a ketone into an aldehyde. Step 6 is the first of the payoff phase. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is catalyzed by G3P dehydrogenase. NAD+ and Pi are required, and NADH and H+ are produced. I'm going to finish glycolysis because it's huge and I don't want to stop it, despite the fact that this video is already over 10 minutes. So we're in the payoff phase. Second step of that is step 7, 1,3-bisphosphoglycerate to 3-phosphoglycerate. This is an energy yielding step which results in the production of 1 ATP for each 3-phosphoglycerate produced. There are 2 ATP that are produced from each glucose. This step is catalyzed by phosphoglycerate kinase. Step 7. This is known as the break-even point, as 2 ATP used in steps 1 and 5 is balanced out by the 2 ATP produced in this step. Step 7. Alright, step 8. 3-phosphoglycerate turns to 2-phosphoglycerate. This step is catalyzed by phosphoglycerate mutase. This enzyme moves the phosphate group from one carbon to another within the molecule. Step 9, 2-phosphoglycerate to phosphoenol pyruvate, facilitated by enolase. Enolase is an enzyme which removes a molecule of water, aka hydratase. Step 10, phosphoenol pyruvate to pyruvate. This is the final step in glycolysis. It's facilitated by pyruvate kinase, which moves a high-energy phosphate from pyruvate. This is the final energy-releasing step. Each phosphoenol pyruvate converted to pyruvate release 1 ATP. Therefore, there are 2 ATP created in this step. As a result, there is a net total of 2 ATP that are produced from 1 glucose entering the glycolytic pathway. 4 ATP produced in step 10 minus the 2 ATP used in steps 1 through 5. I think that is a good place to stop for this video.